this phase contrast microscope. And because of this work, Zernike won a Nobel Prize. This Nobel Prize, it is a prize given if you are able to invent something that is useful to the society. If ever you're able to contribute something to the society. And this contribution of Zernike is really quite useful, specifically in the field of science. You get it? Any question from here? Any question from this side? Okay, so the last part or the last kind of microscope is what we call the This is an example of a phase contrast. Actually, it is also the same as the compound microscope that we have. The only difference is that there is already a built-in light in it. Right? And more detailed parts. Okay. Now, the widely used, and they say the most reliable kind of electron, reliable kind of microscope, is what we call the electron microscope. Electron microscope. So what is the difference of this most reliable, according to many people, it's the most reliable microscope of all. What is remarkable with this kind of microscope? It says that because it operates through electron beams. So it operates through electron beam. So because of this, more detailed image can be seen or can be observed. More parts can be identified because of this electron beam. Compare with other microscopes, compare with the three kinds of microscope, they can operate through the use of or the utilization of light. Compare again with this electron microscope, it uses electron beam to view the specimen. Now, electron microscope, we have two kinds. We have the what we call the TEM, and we have the SEM. Very good. The TEM, or the transition electron microscope, and we have the SEM, the scanning electron microscope. Okay, so aside from this, aside from microscope, scientists or researchers still use other instruments. Why? Because in studying biology or studying this science, we don't only depend on microscope. There are also some researches that need other instruments aside from microscope. So nowadays, an electron microscope. So nowadays, we have these tools. No? We have autoclave, we have centrifuge, we have computers, the widely used computers, we have water bath, and we have jet electrophoresis. I am sure you are not familiar with these terms. Am I correct? Okay. So these are the other tools used in scientific study. So what is autoclave? And how do autoclave look like? It's like a rice cooker or a pressure cooker. Pressure cooker. It is like a pressure cooker. So other tools. First we have autoclave. Autoclave. Actually, autoclave is used primarily for sterilization. Primarily used for sterilization. What do you mean by sterilization? Sterilize. Or it is the complete elimination of microorganisms. Okay. So, autoclave, it is used for cleaning or for sterilizing. Sterilizing the instruments, not only the instruments, the containers that a scientist will be using, as well as the solutions that will be using. Now, how do you think this autoclave works? Do you have any idea how this autoclave works? How this autoclave do its work, its function? Do you have any idea? Autoclave works or do its function whenever it is under extreme temperature. We subject the material to be sterilized under high temperature and under extreme pressure. So these two, the high temperature and extreme pressure, can kill the microorganisms that a scientist needs to eliminate within that thing before he proceeds with the experiment. Okay? So, another tool is what we call the centrifuge. What is this centrifuge? So centrifuge is used to separate solid from liquid part and from other substances that have mixture. So mixture. Yes. To separate the mixture from one component to another. To separate liquid and solid. What else? To separate. It's written there already. Substances of different densities. Meaning mixture. Okay? So how do you think this centrifuge works? Centrifuge. Are you familiar with centrifugal force? Centrifugal force means that there is a spinning action. So within that spinning action, that causes the solid and liquid to separate from each other. This centrifuge is very useful if a scientist is working on a blood sample. If they want to separate the plasma from the red blood cell, the plasma from the white blood cell of an organism. Remember, blood is considered as a mixture. So that's why centrifuge is used to separate this component. Any question from here? Second years? Are there any question up to here? Can I proceed? What is this? Computers. Yes, computer. Computer is considered as one of modern tools. Computers considered as one of the modern tools in the study of science. Why? Because computer nowadays, it does not only store, it even process, what else? And can analyze things. But I guess yes, it can process information, it can store information, it can what? Again, it can process information, it can store information, and it can analyze information gathered. And it is also very useful nowadays with the modern trend of broadbands, the internet connection, the internet access. Scientists have now ways to communicate from each other.